another show that brings you revelatory news from some of our companies doing incredible things. Today with me, I have CEO Amir Reichman from Scinai Immunotherapeutics Limited. They are listed on the NASDAQ exchange with the ticker SCNI. Now, this is a biopharmaceutical company with two complementary business units, one being therapeutic products and two, biological drug development. We're excited, Amir, to hear more about all of that, including your very, very recent news about all the positive momentum and the future growth among key areas. Welcome, Amir. Really excited to have you here.
Thank you very much, Carmel. It's mutual. I'm also excited to be on your show. And thank you for having me here today.
Wonderful. And again, just sending so much appreciation and, thoughts and well wishes. I understand you're coming to us straight from Jerusalem, Israel, and we just send our empathy and well wishes for you and your family during this uneasy and really inhumane time that the war-ridden Israel is undergoing.
Thank you very much. Highly appreciate it.
Of course. So to jump straight into what Scinai Immunotherapeutics is all about, just to give the audience an update, we're looking forward to covering the Q2 financial results that came out very recently, the restructuring deal of the $29 million bank loan with, I believe, EIB, which is the European Investment Bank, which is going to do a lot for equity and see how that impacts shareholders, stockholders. We're looking at how you're planning to return to the NASDAQ compliance, the R&D milestones, so much to cover, including the CDMO division as a revenue growth engine. So firstly, tell us what Scinai Immunotherapeutics does in more detail, and the unique alpaca-derived nano-sized antibody, how this differentiates the company in this highly competitive landscape.
Thank you very much, Carmel. So Scinai, as you mentioned yourself at the beginning, is basically working with two business units, one developing its own innovative pipeline of biological therapeutic products, all based on this unique technology of VHH, single domain antibodies derivative that is originating from alpacas. And the other business unit is the CDMO, where we give R&D services to other small companies. The antibodies are licensed from the Max Planck Institute in Germany and the University Medical Center Göttingen, also in Germany. And the two principal investigators that we work with are Professor Dirk Goerlich and Professor Matthias Doppelstein. Carmel, many times people tell you, it doesn't really matter what was your research topic in your PhD. It's more important who was your supervisor and who was your mentor. And, it sounds like a cliche or a joke, but it's really important to find and collaborate with the best minds, because the probability that their inventions will then materialize to actual commercial products that are safe and efficacious is significantly better. And in this regard, Professor Dirk Goerlich is a world-renowned expert in the discovery and design of antibodies. And so that's why we went there, and why nanobodies versus other things. So in the end, we went for a de-risk strategy where our molecular targets that we are going after are molecular targets that have been already approached by other manufacturers that developed and designed antibodies against them. However, we identified, together with many consulting firms, including LEK, we looked at these targets and said, hey, there is still money on the table here, and there are still significant patient populations that are undertreated. And so we thought, hey, let's take another category of antibodies and take advantage of their physicochemical attributes in order to be able to develop a product that would be a bio-better. So it will still go after the same molecular target, same mechanism of action, same disease, but in a way that will allow us to expand the pie and allow additional patients to enjoy the treatment and open the door for biological treatment for those that cannot benefit from it today.
So just looking at the very recent news that came out and the investment commitment agreement with RK Stone Miami LLC, it's about $2 million. Tell us more about that and how these funds will be utilized to further the company's growth.
Sure. So what Daniel Stone provided to the company is an equity commitment. He didn't buy today shares in the company. And the reason he didn't do that is because the current market valuation of the company is quite small. And we didn't want suddenly, in a $3.7 million market cap, you don't want somebody to throw in 2 million and suddenly buy, 1/3 of the company immediately after the deal, which can impact severely the share price. What we wanted to signal together with Daniel Stone, who is already our main shareholder, is to signal to the community that Daniel believes in the development and growth potential of the company by allowing us an equity commitment. And what does it mean? It means that we are able now to ask for drawdowns whenever we want from now until December 31st in increments of $500,000 a month. So that will provide us the oxygen we need in order to bring forward our R&D plan into the next stage and into the next catalyst, and also provide us with the required working capital in order for us to continue and provide services to our CDMO clients, and more importantly, to expand our outreach with marketing and sales so that we can increase the incoming income of the CDMO. The CDMO is very early stage CDMO. We just opened it less than 7 months ago. And so we see a very high increase in incoming sales orders, but we're not even close to the top potential of the CDMO. And the top potential of the CDMO, as I mentioned yesterday in my webinar for investors, which is recorded on our website, is approximately $24 million a year with an expected EBITDA of 13 to 14 million. But to reach this potential, we need to grow the business. And so the same for the R&D pipeline. The more cash we have now, we will be able to reach the next catalyst in order to increase the appreciation and valuation of the company in the public market, and then next round will be much better.
Fantastic. Now, looking at the terms of the agreement, I'm sure you touched on this very briefly, but the flexibility around the timing and the size of what was issued, how is this going to impact the shareholder structure?
The nice thing here, and that's a big thing to signal to the market about the long-term position of Daniel, is that Daniel agreed to take unregistered shares. So technically, Daniel won't be able to trade these shares into the float for at least 6 months. So for at least 6 months, the company will be receiving cash flows, but without increasing the float and the number of shares traded. That's a big signal and a positive thing for shareholders, because many times companies that are in low market cap, they don't really enjoy the ability with the very high quality investors. They need to go to different hedge funds, etc. And a lot of traders, people that are doing their profit on the trade. And usually these institutes will request warrants these days, 200% warrants, and they'll want everything to be registered, which means that the next day after the deal is done, or a few days later, they'll start to dump the shares into the float and sell them in the market, and they'll keep holding for the warrant to do the upside. But such a deal is creating a massive dilution of the company immediately after the financing. So kind of, on one hand, you get cash, on the other hand, you get diluted. So share price can get impacted here. It's a very favorable deal for the company, and signals to the market the commitment and belief of the single investor in the company.
Sounds like a great deal. Congratulations. And another point I think that we can put some congratulations to is the news about the lead nano AB candidate. That seems really exciting. Can you elaborate on that and the commercial and clinical potential of the program, and also the unmet needs that it aims to address?
Right. So we have two things to talk about here. The first one is the regulatory milestone, and the other one is the scientific achievement. On the regulatory milestone, on June 4th, we met for a scientific advisory meeting with the Paul Ehrlich Institute. The scientific advice meeting is considered an acceptable guidance for the European IND, which is called IMPD filing with the European Medicine Agency, and is also considered to be the comparable of a pre-IND meeting with the FDA. So by going to this scientific advice, we basically showed to the German regulator saying, hey, here's our animal preclinical plan, the CMC, the clinical plan. Tell us what you think. Give us feedback. And we received on July 23rd, we announced that we received a very positive feedback from the PI on our drug development programs towards a phase 1/2A clinical trial in plaque psoriasis. So first of all, they clarified our preclinical toxicology and clinical program for the plaque psoriasis in many aspects. For example, they requested data that will prove that we are actually neutralizing the isoform of IL-17, which is called IL-17F, which is the most abundant cytokine IL-17 form that is in the skin, which of course shows that our drug is going to have a competitive edge with the treatment of plaque psoriasis, which is a skin disease, right? And we demonstrated that. On July 15th, we made an announcement that we received the results from the preclinical trial, and we showed that our drug was substantially effective in neutralizing the IL-17F. But for the advice we got from the PI, they also gave us several breaks. For example, they told us we can go and do toxicology studies using pigs rather than non-human primates. As we know, non-human primates, both, for good reasons, the society is very sensitive to such animal experiments, and so it takes a lot of time to get the approvals, and the costs are skyrocketing, and the availability of the NHPs for such trials is very low. So it gives kind of an advantage to the large, well-financed or large pharma, and small companies, they can get stuck in the queue waiting for such trials. So the PI looked at we basically created a preparation backed with data and evidence, convincing the PI we don't need to do NHPs because of different reasons related to our antibodies. And the PI accepted that and said, fine, you can go do toxicology in pigs, which are much cheaper to achieve, easier to approve, and faster to conduct. The other thing that they approved for us was that we can go and test first in human in people that are already suffering from plaque psoriasis, which means we don't need to go to the healthy persons first. We do the safety checks directly in sick people. So that saves time and cost. You need to do all these preparations and filing and recruiting people. So here we just go directly to people that are suffering from plaque psoriasis. And the very nice thing, we came with a very a good argument and supporting data and information to explain why, specifically in our drug, we can test both the placebo and the drug in the same patient because we are doing local. So it actually makes a lot of sense. If people don't know, psoriatic patients, they have normally symmetric lesions. So if they have, let's say, a lesion on their hand here, they will have almost the same lesion on the other side. So we tested the placebo and the drug on the same patient. So we are going to save lots of time because recruiting less patients, and we will potentially be able to demonstrate both safety and efficacy. Only, not full efficacy, but at least initial signs of efficacy, and do kind of a proof of concept in human beings very shortly, and it's very low costs. So that's a big achievement that we talked about from the regulatory point of view. The other point that you mentioned was on our preclinical trial. And in the preclinical trial, we did a very interesting preclinical experiment. So it's fascinating, and it's a very high level of thinking of the model. And so a professor here from the Israeli Technion Institute, he is the developer of this methodology, and this is how it works. He takes mice that are genetically modified so they will not, have any reaction with human skin. Then we bought or received, donations of skin from people that went through tummy tuck surgeries. And so we took this human skin and then grafted it on the back of these mice. We let the mice absorb, basically get this transplant to, show
Settle
that it's
in.
healthy and not
Right.
rejected, etc. And then after 28 days, when the situation is stabilized, of course, those mice that were not stabilized were removed. But fortunately for us, all the mice succeeded. And then the professor is inducing psoriasis. There is a way to induce psoriasis into human skin, and he induced it. And so then we wait additional couple of weeks until the psoriasis is stable. And at that point, we started to treat. It's basically you are treating a human psoriatic skin, but grafted on the back of a mouse. And then we showed that our drug was very efficacious, not differently from the best-in-class gold standard drugs like betamethasone, which is the topical drug for psoriasis, or succinimab, which is called Cosentyx, which is systemically injected. You might ask, okay, so why bother if you are just similar to others? So what's the difference?
Right.
The point is that let's imagine a person that suffers from psoriasis, which is mild, okay. She needs to come home and she finishes her job. Imagine you come back, God forbid, but let's say that you have this, and then the person comes back and they need to come home and okay, the first thing they need to do? They want to take a shower. They need to put the creams or ointments. And then it starts to affect your life. Okay, I cannot sit on the sofa because I need it to dry. I cannot sleep on my bed sheets because I have the psoriatic lesion on my back. I need to wait until it dries. Person wakes up in the morning, they need to put it twice a day. Okay, I cannot, put makeup or cannot shave. I need to put it, wait until I dry. I cannot put my dress shirt. It sounds small things, but it becomes a disease burden that impacts the quality of life of the person. And that's not the only thing. After a lengthy use, the drug starts to demonstrate skin atrophy. So it generates skin atrophy, and the skin starts to change the thickness. It becomes very thin, and it changes the color. Now, okay, if it's on your elbow, you might not notice too much, but what if it's on the face? What if it's on the sex organs? What if it's on the scalp, palms of hands, bottom of feet? These are very sensitive areas. People don't want to experience skin atrophy in these areas. Imagine you cannot touch anything because the skin is so sensitive. And so we understand the problem with these drugs. The other drugs, which are the biologics that are currently available, they need to be administered once a month. And I'm not joking, that's the cost. $5,400 per syringe per month.
Wow.
So you can imagine that, health funds and all the insurers in the United States and everywhere in the world, they're very sensitive to who they approve the use of such a drug. It's a super expensive drug. And so
it's not easily
normally
accessible to every patient dealing with this. Yeah.
Absolutely not accessible, even for those affluent communities, even in Europe, even in the UK and the United States, not to talk about all the others that were completely neglected by the manufacturers because they cannot afford such a drug. Think about even in the affluent, in the Western world, it's restricted to those that suffer from moderate to severe. And to be honest, most of these people that take it are severe patients. But what about the 75% of the psoriatic patients? They're not severe, but a person that has a psoriatic lesion on her face, on her palms of the hands, on the genitals, they For them, it's the end of the world. They want a relief now, and they don't want to put corticosteroids that will destroy their skin in these sensitive areas. And nobody will give them biologic. And even if they are given biologic, it's super expensive. And so here's exactly where we're coming. And we're saying, we will give you a biologic, but it will be a biologic that you can administer, self-administer directly to the lesion, small amounts. Manufacturing of nanobodies, that's, you ask me what's the difference, and here comes the difference. Very cheap to produce. It's very efficacious. And once it goes to the blood, it disappears. So the effect is staying local. You don't have a full body exposure to the drug. It goes to the blood, 2 hours later, it's out of the kidney.
Thank you. Wow, you really paint a very clear, candid picture for this disease. And obviously there's a rising global burden affecting people with this disease. How to make sure that manufacturing and scalability are, positioned to make sure people are getting the supply reliably, cost effectively. Obviously, all of this is critical. So tell us more about how that plays out for Scinai.
Yeah, that's an excellent question. So to find a solution for your problems, for one's problems, is to be self-aware. 70% of the solution. So we are self-aware. We understand we're a small company. We do not now pretend to say, okay, once we finish phase 3, we will start distributing manufacturing to the whole world. That's not realistic. And that's basically, these are the things and aspirations that get very small companies fail. And we don't have the sales force. We do not have the marketing capabilities. We do not have the ability to bundle products. So the right strategy for us is actually to be the generator of drug development, we are located in the right sweet spot between the universities and research centers to the medium to large size pharma, right? We are developing a pipeline, but we're not developing one drug. We're not a one-trick pony. We're pushing pipeline products all the way from the patenting stage. We're moving them from the academia ways of working to GMP manufacturing, scalable manufacturing, things that you can repeat and do and file, etc. We develop the TPP, the target product profile. We take it through the animal studies, human phase 1 safety, human phase 2 efficacy, and at that point, it needs to be sold to the next step. And then we sub-license it to the larger manufacturer. It can be one for a region, it can be a one worldwide. It depends on the terms, but our strategy is to sub-license, and then we can sub-license the drug for any indication, for a specific indication, because we are not restricted by the use. We can use the drug for one disease, 2 diseases, even 10 diseases. We can do combinations of the drug in a monomer, in one homodimeric. We can use a bispecific, monospecific. So there are many ways that we can play with the molecule to extract value from the molecule. But this is part of licensing out the rights. And with the proceeds that we receive from milestones, upfronts, and of course royalties in the future, we will then go back and start and in-license new products so that we always have, a very nice chain of pipeline products, some of them in late stage, some of them in early stage, some of them in just licensing. So we always progress, and that's how we generate value. In the future, if we decide to become a vertically integrated company that includes manufacturing, supply, etc., we don't know. We might, but that's not the business model as of today.
Makes total sense. You mentioned universities, and I know that the company has established several strategic partnerships. How do these collaborations enhance Scinai's capabilities and also create value for shareholders?
It's an excellent point. Look, many companies look at their research expenses and what is the yield of research. We say we need to do what we excel in doing. What is our best expertise? And our best expertise is drug development, not the research and not the discovery. So we outsource research and discovery. And that's why when we created a collaboration with the universities, we created a 5-year research collaboration agreement with both the University Medical Center Göttingen and Max Planck Research Institute. And so we give them the targets and we say, you know what, we want an antibody for that molecule. Here's the funding, go. And then they basically generate the pipeline, they discover, and we have an acceptance criteria with them. If once they arrive to the acceptance criteria, we can take the option and then we execute an option for an exclusive license worldwide at pre-agreed financial terms. So we don't need to renegotiate everything. Everything is just fill in the name of the molecule. We have a license, and then we take the next one. And so we have this multi-year research. And so they are focused on research, discovery, optimization, etc. Once it is ready to be developed, so upstream manufacturing process development, all the CMC package controls, analytical methods, manufacturing for preclinical, for clinical, that's our sweet spot. This is exactly where we chime in.
Would that mean that there's no real strategies in place to extend beyond autoimmune and infectious diseases at this point?
At this point, no. It's such a wide arena, and we have so many products in our pipeline. And the ability to use the products in different modalities and different routes of administration, different diseases, autoimmune diseases is a wide area. And so for our current size and capabilities, it's more than enough. Of course, with growth, we will probably look at things, but for now, it's fine. It's sufficient for us to. It's also important that somebody generates expertise. You want to be the best in class, right? You want to generate and say, hey, when they think about Scinai, one thinks excellence in antibody discovery, antibody development, so they know they come and they get the best antibody.
Absolutely agree with you on the focus and then getting expertise from that. Let's talk a little bit about ESG now, and how those principles align with Scinai's mission to develop innovative and patient-centric therapies.
We are very, very early stage company. We are looking, for example, the way we manufacture water. We have a very unique water for injection system developed at the Technion. It's a unique system that saves lots of water and a lot of energy because it's a cold system. We don't generate normally to move water for injection in a manufacturing site. You need to keep the water at a very high temperature, and then you cool them down before use. And that's in order to disinfect the water and prevent bacteria and other things from growing in the circulation of water that then need to be injected into humans, right? So because we are in a parenteral industry, we are injecting the drugs. There are technologies today to generate ambient temperature water for injection, and that's a system that was developed here at the Technion. It still keeps the sterilization. It's fully approved by the FDA and EMA, but it saves lots of energy and electricity for us, and of course, CO2 emission, etc. When you generate a drug that is easy to manufacture, easy to transport, for example, one of the nice things about nanobodies, and that's probably something that definitely now it gives me a good idea to mention when we talk about ESG. It's a highly hyperthermostable. You can boil it in 95 Celsius degrees or 200 Fahrenheit without any damage to the molecule. So why is it important? Cold chain temperatures for distribution, warehousing, administration. And so that also impacts the ability to send the drug into different parts of the world without risking of write-offs, without risking of the drug being destroyed, and increasing access to millions of people who currently can't access such a drug. It also increased the comfortability, patient comfortability, because you can put per person can put this small pen for her treatment of psoriasis in the purse or in the bag without being so worried that it will, that it requires refrigeration. And so that's in our small areas. Of course, in the company itself, we have a lot of policies like, no disposable plastics. We don't use, we use only, reusable stuff in our kitchen. In the way we produce, we are using, for example, single-use fermentation systems. It sounds a little bit counterintuitive. People think, oh, single-use, plastic, you need to throw plastic. Indeed. But when you calculate, because when you don't use the plastics, you use stainless steel. Stainless steel require cleaning with caustic soda, which generates a lot of contamination. It requires sterilization with steam, which requires a lot of energy, and then it requires lots and lots of testing. So you have a lot of disposables accumulating in your labs because of testings that you need to take in order to assure that there is no contamination. Here, you use one small plastic bag that then can be responsibly collected and put some, in a specific trash, but not, but you basically saved a lot of all of the others. And there are a lot of articles showing that the balance here is way, way favorable for the single use.
Now, given the company's global aspirations, how is Scinai positioning itself to establish a strong commercial presence in both developed and emerging markets?
Yeah, it's an excellent question. The Max Planck Institute, UMG, and Scinai signed a contract that requires us and made us commit to sell the drugs globally, and specifically in those countries that are designated by the UN as the least affluent countries with people where people live with the very limited means, to sell these products or make them sellable through distributors or through third parties in equitable, we basically say equitable licenses. So we give a specific license locally to ensure that the drug is not going to be available only for those who can afford it, but also for those that hardly can afford such a biological drug.
Scinai Immunotherapeutics Limited is positioned for growth. And to get more updates on their recent developments, please go to Scinai.com. They are also listed on NASDAQ as SCNI. This has been a fantastic conversation. Dr. Reichman, please take care in Israel and all the very best.
That's it for The Harvest, available anywhere you listen to podcasts.